Okay, so let's get started. Good evening and good morning, everyone. So welcome to this time Immune Zoom seminar. So today, with my big pleasure, I would like to introduce today's speaker, Dr. Loke from NIAID, NIH. So before the seminar, I think I'm just give a brief introduction of Peng. So Peng got finished his undergraduate in Oxford, UK, and then continue with his study on parasites immunology in University of Edinburgh, also in UK, to get his PhD. After which he continued to pursue his academia career in UC Berkeley, also in immunology, and then following with another second postdoc with in UCSF, also in parasites immunology. So from 2009, Peng got a position in Department of Medical Parasitology in NYU as assistant professor, started his own independent program. And then from 2009 to 2020, he actually get tenured, become associate professor also in NYU. From 2020, we get really pleasure to recruit Peng to NIH as senior investigator and section chief as the type 2 immunity section in a laboratory of parasitic diseases in NIH. So I think a lot of us probably familiar with Peng's work. His research program is to understand the heterogeneity of type 2 immune responses during parasite infections. And his basic immunology study and then translational research studies on the microbiome are convergent towards to the goal of understanding factors which regulate different type 2 immune response in humans. So Peng got really a different awards and honors. I'm just going to list two. I think he got Pacific Southwestern Regional Center of Excellence Career Development Award from 2007 to 2009. And 2014, he got recognition of scientific excellence in molecular, cellular, and then immunoparasitology from American Society of Tropical Medicine and Hygiene. I think I would just want to highlight one part of his work is, I think Peng is really a very pioneer to study how the helminth infections or the parasite, symbiotic parasite to calibrate the intestinal microbiota for host symbiosis and the host defense during infection. His work really provide different insights for how parasites participate in gut commensalism and the maintenance of intestinal physiology. He really actually provide a lot of high profile publications related to the parasites and microbiome interaction for the mucosal immune response. So without further ado, Peng, please take away. We're looking forward to your talk.
Thank you. Thanks so much for that really nice introduction, John. It's a real pleasure to talk to all of you from so many different places. So as Chuan had said, my interest is in type 2 immunity and helminth infections. And the first thing I'd like to do is to explain to you why I find helminth infections so interesting. Apart from the fact that these are really amazing organisms, you can tell just by these pictures that they are multicellular organisms that are very large, and therefore they're very different from viruses, bacteria, and protozoans, which are the type of infections that most people think about. And these parasites, they don't replicate within the host, and yet they are able to establish these chronic infections. And they're extremely successful because a lot of people worldwide are infected with these worms. It's estimated that somewhere around 2 billion people are infected. And so, during the course of human evolution or mammalian evolution, really, this number is probably closer to 70%, right? So what that means is we have co-evolved with these organisms during our evolution, just like we've co-evolved with the gut microbiota. And in many places throughout the world, people who are infected with helminth are the ones that become co-infected with other pathogens. So for example, HIV, TB, and malaria, but also now coronavirus infection, of course. And we still don't understand the effects that helminth infections will have under these complex situations, as well as the effects that they might have on vaccines and other types of mucosal pathogens. It is important to realize, though, that unlike the gut microbiota, these are not commensals, these are parasites, and they cause pathogenesis, right? But the pathogenesis of these parasites tend to be focused in individuals that either have a very poor immune response, a very weak immune response against the parasites, and then they become super infected and have very heavy parasite burdens and develop pathology because of that, or the host mounts a dysregulated immune response against the parasite that's too strong and leading to immunopathology. So in most people, you have this balance between resistance and tolerance mechanisms that expel some of the parasites, but also tolerate having some of these parasites within the host. And therefore, you can get essentially an asymptomatic infection in most individuals. So really what determines whether these parasites are pathogenic or not is really the variation in immune responses between individuals. And I would argue that this is one aspect of immunology that we have a very poor understanding of, is the inter-individual variation in immune responses within the population. And of course, this variation in immune responses and this kind of curve is a reflection of all infections. I think coronavirus infection is kind of a striking example of this that's in all our minds, where individuals that might have a poor interferon response or overexuberant inflammatory response are the ones that develop severe pathology. And so most immunologists today, and really over the last 20 years, because of the advent of transgenic and knockout mice, have become very reductionist in terms of trying to understand specific pathways in a very detailed cellular and molecular level. But these approaches do not help us to explain variation in terms of variation in immune responses between individuals. And to understand variation, one has to turn to tools that have been developed by epidemiologists and ecologists that study the relative effect sizes, relative contributions of different factors towards the variation in immune responses between individuals. And so this is something that I've become really interested in, and something that especially at the NIH, where one does not have to justify specific hypothesis-driven mechanisms, we can try and develop a research program to try and understand why different people have different types of immune responses. So when it comes to helminth infections, it is type 2 immune responses. The main response is the type 2 immune response that both expels the parasite, but also protects the host from the tissue damage that's driven by the parasite. And what's interesting is this type 2 immune response is also important in tissue homeostasis. And so by understanding helminth infections, we also understand a lot of processes in tissue repair, wound healing, and homeostasis. And so the aspects of the type 2 immune responses are the production of cytokines like IL-4, IL-5, and IL-13, the activation of innate cells like basophils, eosinophils, and mast cells, but also features of immune regulation, because in chronically infected individuals, if you draw blood from these individuals, you find that the T cells are anergic, and there's immune suppression by a combination of regulatory T cells, regulatory B cells, and also alternatively activated macrophages. So these features of chronic helminth infections has led to, over the last many years now, this concept of a role for them in hygiene hypothesis, namely that in a parasite-free population, the immune response curve of the population may be shifted to cause more people to have a dysregulated immune response and increase susceptibility to allergy and autoimmune diseases. So, you know, my training was originally in mouse immunology. I also did, you know, reductionist mechanistic studies. But what drove me to become interested in translational research and human immunology was really an encounter with an individual who had, this was towards the end of my postdoc at UCSF, and I met this individual who had deliberately infected himself with Trichuris trichiura to treat his symptoms of ulcerative colitis. And what was remarkable about his story was that he seemed to be successful at putting his disease into remission. So instead of being in a situation where he was facing a colectomy, he was able to put his disease into remission. And so as we followed him during a period of several years, we were able to obtain biopsies from this individual by colonoscopy and be able to generate a very detailed cellular and molecular profile of events that was happening in his gut during colitis as well as helminth infection. And of course, the problem with characterizing this one individual is that it really is just one individual. And so over the last 10 to 15 years, while I was at 10 years, while I was at NYU, we tried to examine larger numbers of healthy individuals as well as IBD patients to see if some of the features that we originally observed in the first individual were applicable. And so based on these studies, which I won't have time to go into, the model that we developed is that when you have an individual that develops colitis, what you get is expansion of mucosal bacteria that are normally present in healthy individuals, but it's kept in check by both competition with luminal bacteria as well as the mucus layer and the immune response. And these tend to be aerotolerant bacteria, and the role that the oxygen gradient may play some role in leading to the expansion of these bacteria. But when they expand during colitis, you get translocation across the intestinal epithelial cells, leading to the recruitment of Th17 cells and neutrophils into the tissues. But you also get spillage of some of these bacteria out into the lumen. And what we think happens during Trichuris infection, based on what we saw in this individual, but also based on some of the other studies that I'm about to show you, is that you get a type 2 immune response to the parasites that really stimulates the goblet cells to produce more mucus. And this in turn restores the barrier to prevent the translocation of these bacteria across the intestinal epithelial cells. And at the same time may lead to alterations to the composition of the luminal bacteria that can outcompete these inflammatory mucosal bacteria that are present. So of course, our tools have become more sophisticated over time. Initially, we could characterize these biopsy samples mainly by flow cytometry analysis. But now we can apply approaches like single-cell RNA-seq, but also ATAC-seq on biopsy samples that we can collect from individuals during colonoscopies. But really, I think for us, the biggest breakthrough in being able to do more detailed immunology comes from our ability to be able to freeze these biopsies in a way that we can recover cells later on to be able to do analysis. Because when we first started these experiments, we had to do everything with fresh biopsies, and my poor postdocs and technicians would end up, working until 3 o'clock in the morning because they would get these biopsies from the colonoscopy suites in the afternoon or something like that. So once you're able to freeze the biopsies and be able to do everything with frozen biopsies, it really increases our ability to really characterize these samples in many multiple different ways. And one example is this data that we just published recently, where we were able to isolate CD45 positive cells from these biopsies for single-cell RNA-seq. And the interesting thing from the single-cell RNA-seq analysis that we conducted from these ulcerative colitis patients was that we found a population of IL-1B positive, lysozyme positive macrophages that was strongly associated with inflammation, as well as FOXP3 positive, BDF positive T cells. These populations were expanded in both ulcerative colitis patients, but also pouchitis patients. So these are patients that have had their colons removed and a pouch put in place, but then later developed inflammation in the pouch. And so using these biopsies, we've also been able to isolate CD4 positive T cells for ATAC-seq analysis, and we can find enrichment of specific motifs, including BDF, in these T cells, indicating an enrichment for BDF positive cells within this population. So these are some cell types that we're interested in pursuing in the future, and these approaches that we've developed is what we intend to apply towards a trichuris human challenge infection model that we are getting going to here at the NIH. It's really one of the reasons why I moved to the NIH, is because we have access to the clinical center, the ability to conduct clinical trials, and the infrastructure to be able to do so. And so what we've done over the last year or so is to develop a CGMP process to produce trichura eggs to submit an IND application with the FDA, so that we can do challenge studies and really characterize whether or not trichura infection could be safe in healthy volunteers, but then also be therapeutic in ulcerative colitis patients. And at the same time, being able to characterize the mucosal responses, taking the approaches that we have developed over the last few years. So of course, I've mostly discussed some of the immune profiling approaches we've taken, but we think that the microbiota clearly plays a big role in this process. And the first hints of this came quite early on from a study that was done on rhesus macaques by Jana, who was a MD PhD student at the time at UCSF. And what she found was that, that juvenile macaques at primate research centers often develop this idiopathic colitis. And it's one of the main reasons why macaques at primate research centers requires veterinary attention. And the pathophysiology of these juvenile macaques looks more similar to ulcerative colitis than Crohn's disease. And so the vets at the Primate Research Center were very interested in studying this disease, and were able to find 5 monkeys that had this disease, where we gave Trichuris trichuria eggs to, and what we found was that in 4 out of the 5 monkeys, there was an improvement in fecal consistency score. That was basically improvement in diarrhea, whereas there was 1 monkey that didn't respond and eventually lost so much body weight that had to be sacrificed. Whereas in the monkeys that did respond to treatment, they increased their body weight, and as their symptoms improved. And so in this study, we were also able to get biopsies from colonoscopies. And one of the things that we found when we did, this is microarray analysis, not RNA-seq analysis. One of the things we found from microarray analysis was that a lot of antimicrobial peptides and other antibacterial signatures were increased in the colitis, in the monkeys with colitis, but then substantially reduced in the monkeys that responded to treatment. So there was this reduction in antibacterial response after Trichuris trichuria infection. And that was associated with alterations to the mucosal microbiota communities. So in the pre-treatment macaques that had colitis, there was reduced microbial diversity compared to control macaques. And this was, although not significantly increased, was partially restored post Trichuris trichuria infection. And when you look at microbial communities, you can see how, compared to the macaques that had colitis, 3 out of 5 macaques now look more like control macaques. And in 2 other macaques, the populations were also shifted. So this was the first indication to us that maybe Trichuris infection could reverse microbial dysbiosis. And I was very fortunate when at the time that Ken Cadwell also joined NYU, and a graduate student in Ken's lab, Shika Ramanan, was working on the NOD2 knockout mice. And she had found that in our animal facility, these NOD2 knockout mice became colonized by Bacteroides vulgaris. And only in the Bacteroides vulgaris colonized mice do you see increased expression of inflammatory markers like Reg3 beta and increased production of interferon gamma, and also a defect in the goblet cells. So a postdoc in my lab, Roanne Bolcutt, collaborated with Shika to see what would happen when these NOD2 knockout mice were infected with Trichuris muris. And what they found was that Trichuris muris infection restored the number of goblet cells in the villi, and also reversed the increased expression of Reg3 beta in these NOD2 knockout mice. And although the NOD2 knockout mice did not have pathology, they were very sensitive to piroxicam treatment. So they were, compared to wild type mice, when they were given piroxicam, they lost a lot more weight. But if they had been infected with Trichuris muris, they no longer lost weight in response to piroxicam treatment. And so mechanistically, after a lot of experiments, what Shika and Roanne were able to show was that this was actually an indirect effect, whereby Trichuris infection increased the abundance of Clostridialis population, which in turn reduced the abundance of Bacteroides bugs. And basically, this was a type 2 driven mechanism. So loss of STAT6 would ablate this effect, and delivery of IL-13 or IL-4 could promote this effect. And so this was the first indication that changes in microbial populations could affect inflammation and dysbiosis. Now, at the same time, we were collaborating with Yvonne Lim's lab in the University of Malaya. And I'm from Malaysia originally, so it was a great pleasure to be able to collaborate with Yvonne back in Malaysia, who had been working with these orang asli in Malaysia for decades, really, doing field work on the epidemiology of different parasitic infections that affected these indigenous populations in Malaysia. So a graduate student in Yvonne's lab, Su Ching, came to my lab first to look at the microbiota in a cross-sectional study where she had obtained fecal samples from 51 individuals in 3 different villages. And when we performed 16S sequencing on these samples, we found that the helminth positive individuals had a significant but small increase in microbial diversity compared to the helminth negative individuals. So this was really interesting because usually infection is associated with reduction in microbial diversity. But in this case, helminth infection was associated with increase in microbial diversity, just like in the macaque experiments where microbial diversity was restored by helminth infection. So what was also really interesting was that the village in which we performed many of our studies is really only 40 kilometers away from the University of Malaya. And yet the microbiota of these individuals is really very, very different from the urban controls that we looked at that live in Kuala Lumpur. So these differences are almost as great as the differences between North Americans or Europeans as people that live in developing countries. So to follow up on these cross-sectional studies, Su Ching did a longitudinal deworming experiment where she collected stool samples, treated the individuals with albendazole, and then collected stool samples after deworming, and found that indeed after deworming there was a reduction in microbial diversity, and there was also a shift in population where you see increased abundance of Bacteroides and reduced abundance of the Clostridialis groups of organisms that was similar to what we observed in the mouse studies. But of course, there are many other effects, many other things that could affect the microbiota, not just helminth infections. And so Su Ching also did a nutritional survey to look at the diet of these individuals. And we also collected blood samples from these individuals to measure clinical parameters, as well as to perform RNA-seq analysis before and after deworming. And it took us a long time to figure out how to integrate all of these very complex multi-parameter data sets. And eventually it was Mei-san, a postdoc in the lab, who borrowed an approach that was used by Jeroen Reis in his microbiota studies to essentially calculate which of these parameters have the biggest effect size on the variation in the microbiota between individuals. So when she calculated these effect sizes, it became clear that trichuris infection intensity and helminth infection status had the biggest effect size on the microbiota variation between individuals in this analysis that was even greater than dietary fiber, which is something that's well known to contribute to changes in the microbiota. So this approach of calculating relative effect sizes on variation is something that really became important to us as we looked at immune variation between individuals. Before I get to that, I should state that based on these studies, Su-Ching, who is now back in my lab as a postdoc, has embarked on a much larger study on 5 different villages in different parts of Malaysia. So in these previous studies that we described, we worked predominantly in just a single village, which was in Bang Sun, that was close to the University of Malaya. And in this village, 95% of the individuals were infected with helminth infections. And so we thought it was important to determine what happens in different villages that had different prevalences of helminth infection. And so we have now performed shotgun metagenomic analysis on 650 fecal samples collected from these 5 different villages. And one of the challenges that immediately became apparent to us is that most of the sequences that we got back do not map onto the reference sequences in a very easy way. We were able to improve our mapping efficiency by using databases that incorporated much larger sets of MAGs, metagenome assembled genomes. And only by using these MAGs databases can we get decent mapping percentages for all these reads. And additionally, what became clear was that in the different villages, the percentage of mapped reads varies significantly. And places where you have the most helminth infections are places where you have the most unmapped reads. So what this means is that any microbiota genomes that are affected by helminths are the ones that are most uncharacterized. So what we're doing now is we are assembling our own MAGs, metagenome assembled genomes, from this data set in order to be able to have a comprehensive assembly of a comprehensive database of genomes, bacterial genomes that are associated with helminth infected individuals. So that is where we're at the moment when it comes to the microbiota work that is occurring in Malaysia. And so in the last part of the talk, I wanted to tell you about some of our collaborative work that we've been doing with Andrea Graham and Ken Catwell in Princeton, and this is really the enclosure that has been set up by Andrea Graham to look at what happens when we take laboratory mice and rewild them in a more natural environment, this was in a pilot experiment. Jackie, who was actually a technician in my lab, but went on to do her PhD with Andrea, looked at what would happen to laboratory mice that have been introduced to this rewilding environment when they were infected with Trichuris murus. And this was with C. black 6 mice. And what was really interesting, first of all, not surprisingly, the rewilded mice had increased microbial diversity compared to the laboratory mice, which is not surprising considering the change in environment that they undergo. But additionally, what was really interesting was that the mice became more susceptible to Trichuris infection after rewilding. And this was associated with a switch from a Th2 to a Th1 response in the gut after rewilding. But the worms themselves seem to be much bigger and more fully differentiated. And so what this indicates to us is that, so far we have talked primarily about how trichurous parasites may affect the bacterial microbiota, but the microbiota itself, of course, will affect the development and differentiation of the parasites themselves. So work in Richard Grenzis' lab had shown that hatching of the trichurous eggs is very much dependent on the microbiota in the cecum. And so one can easily imagine how alterations to the microbiota could affect the hatching of these worms and their successful colonization. So this is the point I want to make here is that it's likely to be a bidirectional interaction between helminths and the microbiota, and this is something that actually Ken's lab is looking into in much more detail now. But based on the success of that preliminary experiment that Jackie did, and there were a lot of logistical issues that had to be solved in order to do these experiments. In fact, this is regulated by the Princeton IACUC, and initially they made Jackie camp out there because in theory all animal facilities had to be monitored all the time. So they made her camp out there for the entirety of the experiment, which was something like 6 weeks, which was pretty horrible. But Andrea and her lab has now figured out how to put RFID chips into the mice when they're released into the enclosure, so they can be monitored through RFID chip readers, and therefore they no longer have to be camping out in the enclosure during the course of an experiment. So in the summer of 2017, Ken's lab, Andrea's lab, and my lab got together, and we did a large experiment where we examined what would happen to mice that carry IBD susceptibility genes, like the NOD2 knockout mice and the ATG16L1 mutant mice, when they were released into this enclosure. What we were hoping to see was that some of these mice that were susceptible to IBD may develop more severe intestinal inflammation and pathology when they're released to the rewilding enclosure. But actually, we didn't see any signs of increased intestinal inflammation, and the mice were just fine. But this gave us an opportunity to examine the immune response of these mice of different genotypes. And we did so in a number of different ways. But I want to, for the purpose of this talk, I just want to focus on a few things, which is that when we looked at the blood of these mice that have been released into the enclosure by flow cytometry analysis, what we found is that when we take all the flow cytometry data and did a principal component analysis of that complex flow cytometry data to reduce the dimensions of each mouse of the flow data to a single data point for each mouse, what you can see is that the laboratory mice and the rewilded mice are separated out very clearly, and the effect of the environment is greater than the effect of the mice of different genotypes. Now, there is certainly an effect of genotype. So for example, you can see that the NOD2 knockout mice seem to be clustered here. But if you do an effect size calculation the same way that we did for the microbiota experiments I described earlier, you can quantify that the environment has a bigger effect size on the variation of the immune response in these mice than the genotype or sex, because there were both male and female mice that were in this experiment. So what was really interesting was that the biggest change that occurred in the rewilded mice was in the neutrophil population. These were cells that had a high side scatter. What was particularly interesting was that one of the biggest differences between mice and humans is the proportion of neutrophils in the blood, where laboratory mice have a much lower percentage of neutrophils than humans do. And so this is consistent with a more human-like immune response in these rewilded mice. And this increase in neutrophil expansion appears to be associated with increased fungal colonization. And Ken's lab was able to isolate multiple types of fungi from the rewilded mice that were not present in the laboratory mice, and were able to show that the reintroduction of these fungal communities into germ-free mice was able to drive this increased neutrophil abundance. So that is the kind of conclusion that we could draw when we looked at the immune response from the perspective of flow cytometry. Now, if we measured the immune response taking a different approach, where we took the mesenteric lymph node cells from these mice and stimulated them in vitro with different microbial antigens, and then measured the cytokines that were being released after microbial stimulation. Now, what we found there was that, interestingly, the genotype of the mice had a bigger effect size on the response to microbial stimulation in terms of the cytokine production. And you can see this here, where you can see that the NOD2 knockout mice are quite distinct from the other mice. And even though there is, indeed still an effect of the environment, but between the lab mice and the rewilded mice, this effect size is overall reduced compared to the genotype of the mice. So based on these results, we had hypothesized, and this was actually a really elegant sentence, I think, that Andrea came up with, that perhaps the environment, this is our hypothesis, that perhaps the environment is a stronger driver of the composition of the immune system, but genetics may be a stronger driver of the per cell responsiveness. And so this could provide an explanation for how genetics and the environment could combine to determine the variation of the immune response between individuals. And so that's one of the ideas that we are excited to test further, because clearly genetic and environmental factors must interact in a particular way to determine this variation in immune responses. And what we are looking at testing is that, that to test that changes in the environment is more likely to drive composition. So if you examine the immune response through flow cytometry, which many of us do, you might see a bigger effect of the environment. Whereas if you examine the immune response from the perspective of cytokine production after stimulation, and that's when you might see a stronger genetic effect, because that may have a bigger effect on the per cell ability to produce specific cytokines. So to test these ideas further, we've just completed, this summer an experiment, and and this is work from Bola, a new postdoc in the lab, where so the caveat from our previous experiments is really that we were looking at only a few alleles, right? We were only looking at the NOD2 and ATG16L1 alleles. This was not a very wide genetic variation. So what we've done is now use 3 different inbred strains of mice, and that covers quite a lot of genetic variation. We have repeated this experiment where we've introduced these 3 different inbred strains into the lab and into these different environments. But we've also then infected these mice with Trichuris murus. And we ran a flow cytometry panel on the peripheral blood of these mice. And we again reduced the dimensions of the data into this principal component analysis. And what you can see here is the effects of the environment, the genotype, and infection. And if I just walk you through these PCA plots, you can see that along the PC1 axis, there is much greater variation in the rewilded mice compared to the laboratory mice. But there's clearly a very strong effect of the environment on the PC1 axis. But the other thing that we noticed that was really interesting was that along the PC2 axis, so you see in the laboratory environment quite a strong genetic effect. So there's quite a lot of difference in immune profiles in the blood between the different genotypes of mice in the lab. But as they whereas when they're in the rewilding environment, this genetic effect appears to be diminished. So one thing that would be really interesting, if we can think of a way to test this, would be that genetic effects are magnified in a clean environment, whereas in a dirty environment or rewilded environment, genetic differences in immune variation gets reduced. And perhaps this may explain why in settings where you're constantly challenged by microbes, you may be less likely to develop autoimmunity or inflammation, because there's less variation, there's fewer people on the extreme ends of the spectrum that might develop inflammatory responses. Interestingly enough, the infection itself had relatively modest effects on this immune variation at all. So this is just a preliminary look at this data set. We have a lot more to do. We have all the mesenteric lymph node data to analyze. We've performed single cell RNA-seq from some of these samples. So stay tuned, and we'll try and get these results out as soon as we can. Another project that I'll just touch on very briefly before I take questions is what happens when you unwild animals. So this was really came about from a conversation with Jason Brenchley, who told me about these macaques of Morgan Island. So it turns out that there is this island off the coast of South Carolina known as Monkey Island, which has housed rhesus macaques that belong to the NIAID for many years. And so when scientists at the NIAID require macaques for the experiments, they basically trap them on this island and bring them to the NIH for experiments. And so I thought this was really, so you can't do any experiments on the island itself, but I thought this was really interesting because you are taking macaques from essentially a wild environment. They live completely wild on this island, and then you bring them to the NIH, and you basically house them in quarantine for several months before they are used in specific experiments that investigators are interested in. So what we started to do is to characterize some peripheral blood samples from these macaques as early as possible from when they come off the island, and then look at what happened, what the changes to these profiles over time. And what we find that's really interesting is that neutrophil percentages come down, just like neutrophil percentages go up in the rewilding environment. And there's also substantial changes in the lymphocyte populations. And now we're in the process of like characterizing these in a little bit more detail. So I hope I've given you all a flavor of the type of approaches that we are taking towards trying to understand the variation of immune responses between individuals. And we hope to do this through a combination of translational studies in humans and non-human primate populations, but also, taking using inbred strains of mice as an approach to what's introducing genetic variation into these to try and explain effect sizes in this kind of variation. So with that, I'd be happy to take any questions. Yeah, so I have to thank all the people that have been in my labs over the time. This is the first group of lab members that joined me at NYU. And this was the group that we had before we left NYU. And now we have this a new group of people here at the NIH. And we are beginning to be able to get going after the pandemic. So finally, I feel like we are beginning to get some data. So with that, I'd be happy to take any questions.
Okay, thank you so much, Pon. That was super interesting. I think there's some questions. I think Laikwan actually already started typing during your talk. So he asked that because your biopsy was frozen, and then if you actually process for the further sequencing, whether the freezing process will actually lead to missing out some samples. I would assume yes, for sure.
Oh, I'm sure we would miss out some cells that are more fragile than others. What I can, so the only cell type we've looked at so far using that approach is the CD45 positive cells. So we thought out the biopsies and sorted CD45 positive cells for the analysis. So I'm sure the epithelial cells, for example, would probably be much more sensitive to what I do know is that so not in my lab, but in Ken's lab, Ken's lab has been able to grow organoids from these frozen biopsies. So we know that we can recover, we can get enough stem cells to make organoids from frozen biopsies, which is also huge. Because instead of having to do that fresh from the colonoscopy suite, we can freeze biopsies and then do all of those things in big batches, right? I mean, it just becomes logistically much easier to handle.
Have you tried side by side, like frozen resawing samples compared with fresh isolated, to see the sequencing result, whether they're
by single cell RNA-seq?
No, I mean any kind of sequencing. I mean whether the result is comparable.
Yeah, I think we did when we did that originally. We did do some flow cytometry experiments, and they were quite comparable. So the key actually was well, what we found was that it really depends on which fetal calf serum you use to freeze the biopsy. So we actually batch tested. Okay. 5 or 6 different batches of fetal calf serum, and there was a tremendous variation in the recovery success of live cells based on that. So I have no idea what's in the fetal calf serum.
I might gotta ask for some serum.
Yeah, sure. Yeah, I think we do have a reasonable amount frozen off, from the batch that was tested. But yeah, that was the big thing was batch testing the fetal calf serum for freezing.
Okay. I think the other question is, have you investigated why Clostridium transfer can reduce disease phenotype in your trichocereus infections model?
Yeah, that has been difficult to really pin down. So we the two possibilities were that they either induce regulatory T cells, and the regulatory T cells reduce disease phenotype, or that the Clostridia outcompeted the inflammatory bacteria, and both could be, the two mechanisms could work together. I know that a student in Ken's lab tried really hard to show in vitro that the Clostridia that she isolated could outcompete, or could kill the Bacteroides that was causing the inflammation. But it was very difficult to pin down a specific mechanism.
I think you asked also that, do you think it's related to the type 2 immune response?
I think it was, let me try and remember. Well, it's certainly related to the immune response, but I think that the expansion of the Clostridialis was what was driven by the Th2 response, not so much the effect of Clostridium on the disease phenotype or the Bacteroides.
Okay. Stan, you want to go ahead and ask a question yourself?
Oh, food intake. Yeah. So the way it works. Yeah. So
sorry. Go ahead. No, go ahead, Peng, you go ahead.
Okay. Yeah. So just in terms of the food, how it works is that we give the rewilded mice exactly the same food as the lab mice. So like they have access to chow, but they can also supplement, right? So they eat all kinds of stuff that they find, like in the rewilding enclosure, and we don't know what else they eat. I think Andrea, who's more of an ecologist, was interested in that, and she was thinking about doing like PCR and sequencing of the intestinal contents to figure out exactly what was being eaten by the mice when they go into the rewilding enclosure. But yeah, we don't have a good answer for that at the moment.
Okay, I think another question from Sha. You want to ask question yourself, Sha? Okay, I'm going to read the question. So
so we did look at that in our studies, and the difference between the genotypes was actually quite modest. There was a bigger effect, obviously, in terms of the lab and the rewilded mice. There was a big effect of that on the microbiota and the microbiota diversity, but we had a relatively modest difference between let's say the NOD2 knockouts, ATG16L1 knockouts, and wild type mice. So with the latest experiment, the 129 B6 and PWK mice, that we haven't sequenced that microbiota yet.
But I thought you sequencing the wild type mice between the lab and rewilding mice, and you found increased diversity in the rewilding mice, right?
Exactly, right.
But that the level of difference, the between the lab and the rewilding mice in the normal wild type mice, if you actually look at the knockout, is that the same level of diversity difference?
Right. That's right.
Is
There's not a big difference between, let's say, not to knockout and a wild type.
No, I'm just saying that not to knockout in the lab compared with
that's right.
the rewilding. So
Yeah.
the difference not that big.
Yeah, no, the difference is bigger. The difference is big between the environments, but not between the genotypes.
But that's actually is interesting because I remember you saying there's no obvious difference in terms of inflammation, intestinal inflammation.
Yeah, that's right.
But the diversity is increased.
Right.
Okay, I see. Okay, I think I have a few questions. So first I want to actually ask, because at the very beginning, I mean, I remember you're talking about human trichuris infection in terms of modulate the colitis. And then my impression, or I want to get your insight, that do you think that the major effect from the trichuris to the colitis is due to the goblet cells? Because what I understand is the mucus is somehow really different between the infection and non-infection, or there's something else.
Yeah, no, I think that's a hypothesis. I think that's but it's hard to
because you also mentioned that the infection with the parasites also increase, like I said, microbiome diversity, which is more dominant, to the host cell or from the microbiome.
Yeah, I don't know how we can separate those two things out, because I think they are related. Because the yeah, I think that they're related, right? Because the increased mucus production could be what's responsible for the change in microbial diversity.
Is there any way to do trichuris infection on germ-free mice?
Not in germ-free mice, because they need the bacteria to hatch.
Yeah. Okay, yeah, sure.
So that's the problem with and the thing is that in that experiment, it would also be difficult to introduce colitis.
Yeah, that's true. That's true.
So yeah, it's hard to separate out those two possibilities.
And I remember that when you mentioned the Marcus experiment, you mentioned that you're actually looking into the juvenile animals, right? So also with the human infection with trichuris as well, do you think the age, the juvenile macaques actually play a role? I mean, if compared with the older animal?
Well, in the macaques, the reason we use juveniles is just because that's when that's when they develop this colitis. So they develop the colitis when they're weaned from their mothers. So yeah, it was more driven by when they get the disease rather than the infection. But children are definitely more susceptible to trichuris, in an endemic population.
Right. Okay, so the other thing I want to ask is you're actually, I think your technician actually found that the rewilding animal have increased susceptibility to trichuris infections. I want to actually follow that phenomenon to actually ask, do you think, because I think you mentioned in your slide that the rewilding actually somehow convert the immune environment in the intestine from the Th2 dominant to Th1 dominant. Do you think the Th1, because you also mentioned the increased susceptibility, probably because the microbiome in the cecum difference, but do you think the immune response change in terms of Th1 like dimensions shift to make this more susceptible?
Yeah, I think it's exactly the same problem as what we were talking about just now. It's very difficult right now. It's very difficult to separate out those two mechanistically, because it could be the changes to the microbiota causes the changes in the response, in the mucosal response from a Th2 to a Th1 direction, right? So one possibility that we've been looking at is having looking at mice that have defined communities.
Mm-hmm.
So if you can have like a defined wild microbiota community.
But I mean, you can also somehow try to apply the genetic artificial mice, like more Th1 dominant animal, to see whether.
Well, that's a good, yeah, that's true. That's true. That's a good point.
Yeah, I mean, that's also probably can help you. Let's say if a dominant Th1 Th1 dominant animal, one hand can also enhance the susceptibility. On the other hand, you also can check the microbiome composition in your rewilding mice, whether they are actually sort of fit to. I mean, there could be a like bidirectional effect, like microbiome, and then, but if they're not really fit to each other, at least you actually know Th1 definitely can dominate that.
Yeah. That's a good idea.
So I think the last thing I'm actually really find fascinating is the very last statement you mentioned, the environment actually determining the composition of the immune cells. I want to ask you why.
Well, I mean, that's, first of all, it's just a hypothesis, like I think we, but what we always need is to have some kind of hypothesis to test.
Sure.
Why
or let's say how. Let's say, I mean, it sounds like if it's composition difference, either it's can direct.
I guess I'm thinking that, the way we were thinking about it was that the type of cells we have is more affected by, the other point I want to make is that, it's of course relative contribution, right?
Yeah, sure.
It's not like all one and all the other, right?
Yeah, of course.
So it's all, because obviously, trained immunity would affect the amount of cytokines that each individual cell makes and so on. But and that's clearly an environmentally driven effect. But I think it does make sense that how much cytokine a cell can make is hardwired genetically. In terms of its ability to respond and to produce cytokine. Whereas as we age or get exposed to multiple different environments, our bone marrow, stem cells, and different compartments would expand and contract and expand and contract in response to multiple different challenges. And so those kind of cellular, like a cellular set point, may be more environmentally regulated based on exposure, whereas like the ability of each cell to make a certain amount of cytokine when stimulated might be more kind of hardwired genetically. So that's the kind of the idea that we're thinking about right now, basically.
That's really cool. I mean, I think I'm really looking forward to the macaques, de-wilding experiment. I mean, that's going to be really exciting.
Oh, good. Okay. I'm glad.
Yeah, I mean, I would like to hear more about it. So if nobody else have questions, I want to thank again Pom for really exciting, fascinating work. it's really a lot of things combined into this talk. I think a lot of interesting things, but really appreciate that you're doing this for us. I think we're looking forward to the development of your study.
Yeah, thank you very
Thank
much.
you so much.
Okay.
Yeah.
Bye.
Good evening. Good night, everyone. See you. Bye-bye.